Belušič from Biotechnical Faculty as well.
Hello, everyone.
Okay, we can see you. We can also hear you.
Yeah,
please.
Thank you for inviting me.
The floor is yours.
Yes, thank you. So our study that was awarded by the research agency and the University of Ljubljana is a study on polarization vision on horseflies. These are quite exceptional insects in terms that they use polarization vision, detection of polarized light for the detection of their targets, their prey, and also water bodies and also navigation. So target detection is performed by seeing the horizontally polarized reflections from mammalian fur. And if we observe a shiny horse through polarized sunglasses, you can see that under a horizontal angle, the horse appears shiny, and under a vertical angle, the horse will appear as non-shiny. And instead of rotating sunglasses, horseflies are equipped with photoreceptive cells that are sensitive either to horizontally or vertically polarized light. We know that because we found it, of course. This is quite frequently found in other insects, but horseflies are like specialized for this, and they have a prominent behavior. And these photoreceptors occur in discrete optical units of the insect compound eye called the ommatidia. Each unit is an equivalent to a camera pixel and harbors photoreceptors that convey signals to the channel for achromatic vision, color vision, and, as we learned, also polarization vision. Next slide, please. Next slide, please. Okay. So we used first the anatomical analysis of horsefly retina to identify the candidate cells that could be used for polarization vision. And we used serial sectioning with a special serial block face scanning electron microscope to find cells that have aligned and straight light sensing organelles and do not perform a twist, such as we see on the left side panel. Receptors are marked with R1 to R6, and we see that one pair of receptors, R7H and R8H, have straight light sensing organelles, and everything else is rotated. So the straight pair is the suitable candidate for detection of polarized light. Then we used microelectrode recordings to characterize the spectral sensitivity of these cells, and we found that the polarization channel only works in the ultraviolet and blue channel, while ultraviolet and green sensitive cells form pairs for the detection of colors. And then we went to the field and we attracted horseflies with shiny beach balls that are spherical approximations of horses and cows, and then we suspended the balls below color filters, and we found that if we eliminate UV and blue light, the horseflies were equally not interested as if the balls were not shiny, as is the case with the matte ball. If we suspended the balls below blue filters, the horseflies got extremely excited, because we know that we enhance the contrast in the polarization analyzer pair, and we also abolished green light, which acts as repelling. Next slide, please. Former slide, please. Yes, thank you. So our research impact of the study is, first of all, that we explain the neural basis of animal behavior. We know how the horsefly attack is triggered. They must observe a dark object. They must see the reflection in the ultraviolet and blue channel, and the object should not be reflecting green light, which helps, obviously, horseflies to discriminate shiny horses or cows from shiny leaves. Those cells are situated in a special segregated class of ommatidia that form, together with other ommatidia, the so-called retinal mosaic, which is a stochastic system, which is a model system in developmental neurobiology, very similar to the one found in the model animal fruit fly. So the findings have implications for developmental neurobiology in general. We also explained the mechanism of color trapping. We know that horseflies are extremely attracted to blue, which does not reflect UV, and an equal situation is found in traps used for tsetse flies, which are much more dangerous than for horseflies. And we know how to optimize those traps and make super traps. And then the retina is also a blueprint for a hybrid imaging sensor, so we know how the horseflies approximately see their targets in the polarimetric channel in the UV and blue, not in the green. And equally, we can use a hybrid sensor to detect, for example, cars that are also polarized objects, even if the camera is optically degraded, such as is the case with horsefly retina, which has a very low resolution. And along with the study, we developed a new stimulation system, which was successfully applied for the detection of color blindness in humans. So a stimulation system that was used for horsefly characterization equally well performs in detecting human subjects without mid or long wavelength sensitive cones. And now we are developing this system together with the eye clinic hospital for the clinical testing in an objective manner. Thank you for your attention. Questions, welcome.
Okay, thank you very much. Do we have any questions? Milos Shinkovets, you have a question? Mr. Shinkovets?